I'm Deva.
Hi, I'm Daya.
We are from D&D Films. Today, we are going to share with you an interesting topic. Enjoy! How do you could relate these features? I'm very sure there are some ideas might be popping in your mind. Here we go. So now, let's get further into this. Inter-individual variability is an important factor that influences drug efficacy and drug toxicity. There are different causes of variability in drug response. First, inhibition of drug metabolizing enzymes or transporters. Second, co-administration of other drugs. Third, genetic variation in the metabolizer or transporter gene. In this video, we will focus more on the third cause. This is the definition of polymorphic variant, and there are three groups of metabolizer as you can see below. Here is the main outline for this video. Drug metabolizing enzyme consists of cytochrome P450 and UDP glucuronidyltransferase UGT, while some examples of drug transporter proteins are organic anion transporter OAT, ATP binding cassette ABC, and multi-drug toxin extrusion MATE. Gene expression of both DME and DTP are regulated mainly by orphan nuclear receptor such as pregnein X receptor PXR, constitutive androstenedione receptor CAR, and aride hydrocarbon receptor ARH. This regulation will eventually affect the metabolism and response of drug differently in each individual. Now, let's see the different variants of DME. Firstly, the cytochrome P450. It is mainly located in the endoplasmic reticulum and highly distributed in the liver. It catalyzes the initial step in oxidative metabolism of endogenous and exogenous substances. Here is how these variants are being classified based on the protein sequence. The examples of these variants are as below. The next example of DME is UGT. It is also mainly located in the endoplasmic reticulum and highly distributed in the liver. It acts as a clearance mechanism or detoxifying agent for drugs and endogenous compounds through a process known as glucuronidation. The UGT variants are classified based on their evolutionary divergence. UGT genes are included in 1A, 2A, and 2B subfamilies. The examples are as below.
Drug transporter proteins consist of organic anion transporter OAT, ATP binding cassette ABC, and multi-drug and toxin extrusion MATE. Details about all these transporter proteins are listed as below. types of orphan nuclear receptors are pregnein X receptor PXR, constitutive androstenedione receptor CAR, and aryl hydrocarbon receptor ARH. Details regarding all receptors are listed as below. As PXR is activated by ligand, it will form a heterodimer with retinoid X receptor RXR alpha. This heterodimer will then bind to xenobiotic responsive element XRE in 5' region of target gene through DNA binding domain (DBD). Thus, regulation of CYP metabolism is achieved through gene expression. As for CAR, the mechanism for regulation of CYP gene expression is similar to PXR. The only difference is that in this case, CAR is the one that forms a heterodimer with RXR alpha. Finally, for AHR, there is a slight difference in its mechanism compared to these previous two. Instead of RXR alpha, AHR forms a heterodimer with nuclear translocator protein, ARNP. In summary, we have already discussed all the topics in our outline. Thank you and good day!